Hello students. Today we will study the next experiments on respiration. In first part of that experiments you have studied that experiment to show that carbon dioxide is produced during the respiration. Now in these next experiment we are going to study to show that the oxygen is used up in the respiration and to show that heat is evolved during the respiration, and at last we will demonstrate anaerobic respiration in the germinating seeds. So let me start with this experiment. Oxygen is used. So here we are taking two test tubes, test tube X and test tube Y. We will label them test tube X and test tube Y. In test tube X and Y, we are filling it with the help of potassium hydroxide solution. So KH solution is poured in both of the test tube and placing that wire mesh, and on the wire mesh we are placing the germinating seeds in the test tube X, while the boiled seeds in the disinfectant in test tube Y. So now the question is arising here that why we are placing the boiled seeds here. So boiled pea seeds we are placing here because they are the dead cells, they will not show the respiration, and this is the control setup and this is the experimental setup. What is the difference between the experimental and control setup? In experimental setup we study a condition and the influence of the different factors on that condition. So here we are studying the process of respiration and the effect of the different that factors like oxygen is used up in the respiration. So we are going to study this the use of oxygen. Here we are placing or making this control setup in order to compare it with the experimental setup to prove that some changes are taking place here, not in the control setup. So this control setup is just for the comparison. In control setup we are maintaining everything similar to the experiment, but the condition which we are going to study in the experiment will not be there in the control. So boiled peas here we are placing, which are soaked in the disinfectant. So now the second question arises that why are we using the disinfectant here? So disinfectant are the solutions in which these seeds are washed in order to avoid any kind of fungal or bacterial growth. So they may interfere with our result. Now after placing the seeds, we will connect both the test tube with the help of the delivery tube. You can see here the tubing 1 and the tubing 2, and both the delivery tube further they are placed in the colored water filled in the beaker. So the colored water also will here help us to see or to check the level of water in a easier way, because water is being colorless, so it will help us to measure the level of water in a easier way. When the color water will rise up, we can identify it in a easier way than the colorless water. So colored water we are using. Now after this we will seal the mouth of the test tube with the help of cork. So now the whole setup is ready. Now leave this setup for few days. Now after that what shall we observe? So before we leave it for the few days, one more thing students you need to do, that have the first reading of the level of water in both the delivery tube. So we can compare the level of that water after the 3 to 4 hours. So first level of the water we have noted down, then we have left the setup for 3 to 4 days. Now what shall we see there? The level of water in this delivery tube 1 here, it shows a greater rise in the water level than the water level in the tubing 2. So we will study the reason for this. Why does it happen? And the second question, how do we know that the oxygen is used up in the respiration? So that also I will answer. So the first question, there is rise in the level of water. क्यों rise हुआ है टेस्ट ट्यूब यहाँ पर delivery tube 1 में water का level? Because you know children that the KOH, it absorbs the carbon dioxide. So there will be no availability of the carbon dioxide which is actually released by the germinating seeds during respiration. And you know, the germinating seeds during respiration, they have used the oxygen also. So here what is happening, the volume of the air is decreasing, and it creates further the vacuum in the delivery tube. So in order to fill that vacuum, water rises up more in the tubing 1 than the tubing 2. Here the boiled seeds are there, as such no respiration, so that much level of that water will not rise here. This is the answer of first question. Now we are going to study the aim of the experiment, that oxygen is used up. So to check it, we will introduce a burning paper into each test tube after removing the cork. So we will remove the cork and we will introduce a burning paper in the test tube X also and test tube Y also. So what will you find here? You will find that flame is immediately put off in the test tube X. Okay, the burning paper will put off here, but it continues for a short while in the test tube Y. Again question. Okay, so why it is happening that test tube X के अंदर जो burning paper है, it puts off immediately. Okay, it's flame. So reason is there because you know that burning requires oxygen, and no oxygen is available here in the test tube X because it is utilized already in the germinating seeds by in the process of respiration. But here in the test tube Y, the availability of the oxygen is there because the boiled seeds, they do not undergo respiration, so it burns for a short while. So this shows that there is a oxygen used up in the process of respiration. So understood, children? The various questions are arising from here, from this setup, how and why. Now next, heat is evolved during the respiration. So for this, we are taking 2 thermos flask. So now instead of that simple glass test tubes or the flask, we are using the thermos flask. So again the question arises, why do we use the thermos flask here? So we are using the thermos flask because they are made up of the insulating material, heat insulators. जो इनकी walls होती है ना, वो heat insulating material की बनी हुई होती है. So that does not allow that the heat of that content or the substance placed in the thermos flask to escape out, and neither it is being affected by the external change in the temperature. So we are using this 2 thermos flask. Now taking some of the soaked seed. So we are placing some seeds for 24 hours in water. They are the soaked seed. So equal number of soaked seed we are taking, and 1/2 of the that seed we are going to boil it. So in first this flask, let me label it as A, and this is the B. So हम A flask के अंदर germinating seeds को रख रहे हैं, like germinating pea seed suppose we are taking, and here we will boil the soaked seed and placing here the boiled seeds, and these boiled pea seeds, they are the dead one. Now the boiled seeds, we will wash them with the dilute carbolic acid, so they are treated with the disinfectant to prevent any bacterial growth. Okay, now here we are placing the live seeds, germinating seeds in A, and in the B we are placing the dead seeds. After this, we are inserting the thermometers in order to measure the change in the temperature. So placing the thermometers, and then after that we seal the mouth of both the flasks with the help of cotton wool, and we will take the first reading of the thermometers in both the flask A and B. Now we will leave each of the flask for 3 to 4 hours. Now after 3 to 4 hours, we will see or measure the temperature here through the thermometer. We'll see, is there any change? Yes. You will find that the temperature in the flask B does not show any rise, while the thermometer which is placed in this flask A shows a higher reading. Why it is there? Because here the seeds are germinating, and germinating seeds, they are producing the heat, and that heat is rising the temperature in the this flask A. So what does it show? It indicates that the germinating seeds gives out heat during the respiration. That was the aim of our experiment. So we concluded that the heat is evolved during the respiration. Now the next experiment to demonstrate anaerobic respiration in germinating seeds. So children, you know that anaerobic respiration means the respiration in which oxygen is not involved, taking place in the absence of oxygen. So here what we are doing, we are showing that the germinating seeds can respire even in the total absence of air. अगर air की availability नहीं है तब भी germinating seeds जो हैं वो respiration को show कर रहे हैं. So that is the anaerobic respiration. So for this we are taking 4 to 6 soaked seeds and remove their testa, means the outer covering. हम 4 से 6 seeds लेंगे. Soaked seed means placed in the water for 24 hours and we will remove the outer covering testa. Peeling the testa from the soaked seed will increase the diffusion of carbon dioxide from the seeds. So easily जो है ना diffusion gases का हो पाएगा, carbon dioxide easily release हो पाएगी seed से. So इसका diffusion increase हो पाएगा. So that is means that will be easier. So that is why we are removing the outer covering. Now introduce these seeds in a test tube filled with the mercury. You see here the mercury is filled in both the test tubes and ensure that the seeds float in the mercury at the top of the level. So seeds are floating, being lighter in the weight, okay, than the mercury, so they will float here at the top level. Now this becomes the experimental setup A. This we will label it A here, the test tube A, and leave this experimental setup for about 48 hours and then observe. And for the control experiment, what we are doing, for the control experiment, instead of this germinating seed, we will use the boiled seed. Till now what we did the same, कि हम control setup में हमेशा boiled seeds ले रहे हैं. So the control setup also we will use another setup here, and instead of germinating seed we are using the boiled seed. That will be the control setup. And here you will see the beaker is also filled with the mercury. Okay. Now after this, what shall we observe after 48 hours? What shall we observe in the test tube A after 48 hours here, or the 2 to 3 days? You will see here that the level of the mercury falls. Okay, so here you will see the mercury is filled till the top of the test tube, but after 2 to 3 days or 48 hours, what shall you observe? There is decrease in the level, fall in the level of the mercury in this test tube. Why? This is because the carbon dioxide gas liberated from the pea seeds has pushed the mercury down. So carbon dioxide is being accumulated here. Okay, now we will see it further. Taking some moist pellets of potassium hydroxide and introduce them into the test tube A. थोड़े से हम कैल्शियम हाइड्रोक्साइड के pellets लेंगे और test tube A के अंदर enter करेंगे. After some time you will observe that the level of the mercury rises almost to the top. अगर हमने यहाँ पर test tube में KOH के pellets डाले तो क्या होगा कि जो लेवल है mercury का फिर से rise हो जाएगा. Why? Because potassium hydroxide absorbs the carbon dioxide gas. So no change means will be there in the control setup, but there will be change in the experimental setup. So we have proved that the anaerobic means we have proved also that carbon dioxide is produced here, and also we have demonstrated this is the anaerobic respiration in the germinating seeds. Without oxygen it is taking place because no availability of the oxygen here in this setup. Only seeds are surrounded by the mercury. So students understood. So all these experiments of the respiration are over, and you study these experiments from the book also, solve the back exercises and ask your doubts.